I'm Heather Brown. I'm the Vice President of Scientific and Medical Affairs at Cord Blood Registry. CBR is so pleased to be a sponsor of this premier stem cell event, including this panel, Banking on Cord Blood Stem Cells is the Future Here. As a pioneer in stem cell collection and cryopreservation, CBR believes in the power of newborn stem cells, not only to improve lives today, but really for their potential to revolutionize healthcare. Between our corporate offices in the San Francisco Bay Area and our 80,000 square foot laboratory and storage facility in Tucson, Arizona, which houses over 500,000 umbilical cord and cord tissue stem cell units, we are the largest and most technologically advanced newborn stem cell bank. We've also made significant investments in advancing the clinical applications of umbilical cord blood stem cells by partnering with researchers to establish FDA-regulated clinical trials, and that's what we're going to talk about today. With all these new emerging investigational regenerative therapies using umbilical cord blood stem cells underway, what do we know now? What do we think about the future? For the discussion, we have a very distinguished panel of experts. After I introduce each expert, they will give a brief presentation, and then we'll hold all questions and discussions to the very end after each panelist has had an opportunity to share their perspectives with us. Our first panelist, I am sure, needs no introduction to many of you in the room, but I'm just going to go ahead and read some of his bio because it's just so darn impressive. Dr. Mahendra Rao has worked in the stem cell field for more than 20 years with stints in academia, government, and industry. He's the Vice President of Clinical Strategy for Q Therapeutics and the Vice President for Regenerative Medicine at the New York Stem Cell Research Institute. Previously, Dr. Rao served as the founding director of the National Institutes of Health Center for Regenerative Medicine. Prior to his period of service at the NIH, Dr. Rao spent 6 years as the Vice President of Regenerative Medicine at Life Technologies, after he served as the Chief of Neuroscience at the National Institutes on Aging and co-founding Q Therapeutics, a neural stem cell company in Utah. Dr. Rao served as chair, member, and industry liaison for the Cell Therapy and Gene Therapy Advisory Committee of CBER and serves on the advisory boards of many companies. Dr. Rao received his medical degree from Bombay University in India, his PhD in developmental neurobiology from the California Institute of Technology. He has an extensive background on teaching medical and graduate students, and hopefully will teach all of us something here today. He's published more than 300 papers on stem cell research and really is internationally recognized for his achievements in the stem cell field and neuroscience research. So please join me in welcoming Dr. Rao. Thank you.
And I should add that it's also a real pleasure to be on this panel with Charles and with Francis because they've been pioneers in their own way in working on cell-based therapy. So what my goal today is to try and tell you about one aspect of cord blood banking and not the main business of cord blood banking, but really what's related to how it can be used for new aspects of regenerative medicine. So quick background, you have umbilical cord stem cells, and I want to make sure that people are clear that there's more than one type of stem cell that's present in the cord. You've got the blood stem cells that are present there, and that's what we talk about when we talk about cord blood banking. You have other blood progenitors like the endothelial cell progenitor, the EPC, that's present there, and you have some additional MSC-like cells that are present in the blood. And then you have the placenta itself, which is a source of unique types of cells, and you have the cord, which is a source of additional types of cells. Cord blood samples have been used in cell transplant since the 1980s, and mostly they've been used as a substitute for marrow-derived blood stem cells. You can imagine that the single commonest use of cord blood-derived blood stem cells has been as a replacement of your endogenous hematopoietic stem cell that's present in the marrow, and that replacement can be for a wide variety of issues. It could be because of cancer, it could be non-cancer related diseases, and it could be because you're trying to use non-CD34 positive cells, which are still blood derived, but the late progenitors as a replacement during the process of recovery in bone marrow transplants. Okay, if I can go back here. I want to make one important point here that you'll see right here on this slide, and that is use for iPSC. And what I'm going to try and focus on is using cord blood derived cells for making induced pluripotent stem cells and why that might be a logical way to go. Couple of important background points that you want to remember is that cord blood transplants have been increasing in number, but it's not been a very steep increase in terms of what can be done. And there are just two simple reasons why that is the case. One, it's increasing because you can collect and there are large number of samples that are present in different banks. And it's clear that cord blood transplants can survive with one or two mismatches, which is a little bit more than what you can do with bone marrow transplants. So that's been a reason for the increase, right, in being able to do it. The reason the increase has not been as steep as one would like has simply been because the cost of a cord blood transplant is a little bit more than the cost of doing a bone marrow transplant when you source cells which are matched. Okay. So you can see that there's been a slight downturn, therefore, in collection in private banking, and the reason for that has simply been because the economies have been slow. Okay. So this is the standard process by which you go through this, and I want to remind you what I said about the cost. By contrast, the cost of a bone marrow transplant is about $8,000 or $10,000. So that's the difference that you have to live with, and you have to understand that the cord blood sample is not sufficient to treat more than an individual of birth of a weight which would be about that of a teenager. Okay, so just keep that in mind in being able to use a single cord transplant. There's another big problem which people don't talk about in terms of cord blood transplants is that historically the FDA has said pooling of samples is not allowed. Okay. So even though you had 2 cord blood units, pooling them for therapy is not considered reasonable medicine yet. And this was a historical thing, and you have to get an exemption before you do that, and the University of Minnesota has moved forward with that quite aggressively in being able to do it, but it's a law. So we have to worry about the law, and we have to worry about whether that's applicable for some of the uses that we think about in terms of pooling. So 3 things to keep in mind. Cord blood collections have been increasing, usage has been increasing, collections have been declining a little bit. The use has been limited because of cost, even though there's a preference because of mismatch that can be tolerated. Okay. I'll skip this slide except to make one point, and that is a point for using cord blood derived products for non-cord blood, standard traditional cord blood uses. Okay. So standard cord blood is collected in a bag. It's collected with a minimal amount of preparation, and it's generally stored with a cryopreservative. What that means is that if you store everything in one bag, then you have to thaw out the whole sample because refreezing a sample is difficult. It changes the viability, and and it also changes it as a definition of a product that you can use in clinical use. So there's been some changes in storage so that you have multi-bag storage in case you want to use a fraction of the cord. And you also always keep some tails in the tubing, which you can then use for testing should additional testing be required. And those 2 pieces are going to be very important when I consider additional uses of a cord. Okay. So 2 models have come forth in terms of using cord blood banking. And this is dependent on knowing a little bit about the immune system. So I'm going to repeat that, and for the experts, I apologize because I'm going to simplify a whole lot of things here. So a lot of people have felt that if they use their own cord and, own sample for therapy, then that will have potential preference versus using an HLA match sample. Now people have argued, why should there be that difference and why should there be that preference? In blood, we don't think that that's the case. And the reason why with cord or bone marrow transplants this is the case is familial transplants have always done better than HLA matched transplants. And the reason for this is the immune response is not just because of the major HLA type, but it's also because of minor HLA antigens which we can't type for because they're very variable. So historically, if you follow the rates of transplants, you will find that familial transplants do better than doing HLA matched transplants even if you do a 10 out of 10 matching, right? So that has led to the idea that if you had a preference that you'd have to have less immune suppression, in the long term it would be less expensive if you could get that done. And that's what's led to the idea that you should have private banking as an option for people who can afford it in being able to move things forward. Okay. So public banks provide you with cords. Those cords are HLA matched, HLA matching allows you to do transplants successfully in non-related recipients, but you have to immune suppress. That amount of immune suppression and the incidence of graft versus host disease is a little bit higher than if you were to do autologous transplants in the same individual, or you were to do familial transplants, or if you were to use cord samples in a private banking model. Okay. So now skipping forward to doing this to a second piece that I want you to remember here, and that is regulations. Okay. So cord blood banking has been in existence, as you know, for more than a decade, maybe 15, 20 years now. And in each case, it was treated as practice of medicine. Even though you stored the cord or you stored bone marrow, it was considered a procedure. And a procedure was much like doing organ transplants. And therefore, the FDA was not involved in any of this as a regulated product. It was involved in test tissue processing at the risk of transmission of diseases because you were transplanting in another individual. So that's a very important thing to remember. So all of the banking efforts were designed just to follow good tissue practices and then as a surgical procedure be transplanted in a variety of indications, and you went to your IRB for a clinical trial for what you're going to do in bone marrow transplants. After 2007, the FDA changed the rules and has said that it will be regulated as a product for at least the public cord blood banks. And so 5 of the cord blood banks have had to go and get a license for cord blood banking. They've also defined what will be a product, which means that if you process it, if it's more than minimally processed, then it will be regulated as a product. So if you are using it for a non-homologous use, so if you are taking cord blood and using it for the heart or using it for, critical limb ischemia, it will now be regulated as a product because it's non-homologous use. If you expand the cells because you now want expanded hematopoietic stem cells and you're doing it for bone marrow transplant, even if it's in the same individual and it's an autologous product, it will now be considered a product. Okay. So the FDA narrowed what would be practice of medicine and expanded what would be a product. And what that meant is that many of the uses that we've been thinking about as expansion of the uses of cord can't be done under previous regulations, or with the assumption that this would all be under good tissue practices. It's also clear that other countries are moving along the same line. So this is important. So it's happened already in the United States. The public cord blood banks have had to get licensed. It's not so true currently in Europe or in Japan or in Korea, but it looks to us that the regulations are going to change along the same lines. There's also this issue that has come up in a legitimate way, that even the private cord blood banks, even if it's autologous use, if they're more than minimally manipulated and it's non-homologous use, right, those two definitions that I added, it will mean that the cord blood banks will also, private cord blood banks will also have to get a license. It's not yet clear because that's not been applied by the FDA, but that's something that you have to worry about in terms of planning and strategy in terms of cord blood. Okay. So that means everybody needs to know about the FDA and how it regulates things. Okay. So that's what you have here. There are 4 parts to regulation. The first part is just what we just heard about, right? So cord blood was not regulated under Section 21 CFR 1271. It was considered a practice of medicine. It was a surgical procedure. And so you didn't need to go to the FDA other than for tissue practices and viral testing, etc., to get a license for a process. There are these 3 pathways. This is the most complicated pathway. This is the less complicated pathway in which you have autologous use, but you still have to get a license from the FDA. And this is sort of the easiest pathway, which was for devices. So companies like Medtronic, which made stents, could go to the FDA for a product that was implanted in humans and then get a license for the device, right? And there were class 1, class 2, class 3 devices, but it was a much simpler rule. And again, just for simplifying things, let's just put years and numbers on this. So if you wanted to get approval from the FDA for a device, it normally took you between 6 months, 9 months, at most 18 months to be able to get that. If it was under 361, then it started with being 2 years, 3 years, or 4 years to be able to do it. And if it's under Section 351, then generally the rule of thumb has been it's 4 years, 5 years, 8 years to get approval, okay, through the FDA process when you get through things. So keep those numbers in mind. So it's really important for cord blood banking and for use of cord blood to now say we have to know whether it's going to be regulated by the FDA or not. And if it's going to be regulated by the FDA, under which section of the regulations will it be regulated? Because that will predict how much time it will take. Of course, I'm giving you average times, it's very possible that you can get fast track status, or you can get orphan disease status and may be able to get that, or you can get research use exemption or compassionate use exemption, which are all different categories, but that would be your basic pathway as you move forward. Okay. And so this is what I'm going to remind you of. So this is section 351. If you were to move forward, this is how you would move forward in being able to get through the regulatory process. And the only important thing I want you to remember from this slide is what the FDA tells everyone. And that is, there are many opportunities to meet with the FDA. And it's really important to use those opportunities to talk to the FDA, because the FDA is not your enemy. And it's not there that they're standing there saying, how do we reject things? They're there to try and help you accelerate the process, because for them, success is number of products that they've approved, because they charge you a fee for being able to approve a product. Right? So that's a measure of government success for them. And so they have formal meetings and informal meetings. And in regenerative medicine, they've been very open to that. And I would encourage everybody to consider that. And the reason for considering that is that it's like going skiing downhill, right? Once you've made a commitment to a path, it's very hard to change without losing time and money. And so if you make the wrong decision or you haven't gotten feedback early, you're going to be committed to that path. And it could be as simple as, I didn't take the right consent, but I went forward and I got great data. You don't have the right consent, you're not going to get approval, right? So consent was done in the first month when you started getting a tissue sample before you started down the path. So that's why it's so critical to have that kind of conversation and make sure you know exactly where you are and what the rules are in being able to get that moving forward. So these are the considerations as I pointed out. So I'm going to just summarize what I've just said so far. As a business in cord blood, you have to make one very important decision. Is the therapy or the sample that I'm using going to be used in a way that's going to be regulated by the FDA or not regulated by the FDA? Once that critical decision is made, then you have to make the second decision. Under which category will this be regulated? Will it be regulated because I'm just supplying input material for a final product? Is it going to be regulated as a device? Is it going to be regulated for homologous use, but in a non-autologous fashion, but I'm going to do minimal processing, etc. But once you've done that, you will see what the time periods are. And I want to remind you that if it's the standard process for under Section 351, it's going to take you 4 to 8 years. Okay. So now I'm going to switch gears a little bit. So now you know what's going to happen. This is the standard sort of process with which cord blood is used. Why in normal process of cord blood usage, what's happening in terms of the business market, why people still prefer private cord to public cord for certain indications as long as it's available because of immune matching issues and minor antigen compatibility and graft versus host disease. And I told you that there are additional uses, all of which will be regulated under the FDA. So now I'm going to go and talk to you about one particular use of cord blood. And that particular use of cord blood is simply using it as a source material for iPSC work. So what are iPSC? So iPSC are induced pluripotent cells, and they differ from the stem cell that's present in the cord in one very simple characteristic. The induced pluripotent stem cell is like an embryonic stem cell, except it's not derived from a fertilized egg, and it can be used to make any cell in your body. There are 212 described sort of tissue cells present in the adult, and each one of them has been shown to be made from iPS cells. So people have been very excited by this idea that we could take iPS cells, use them as starting material, as long as you could generate them from an individual, and make all of these tissues or organs or cells, and then use them for particular therapy. So the question then has become, how do we do it? And there are 3 models that people have talked about. One model is very similar to what you think about in terms of private cord blood banking, is you take some cell or tissue from an individual, and you bank that tissue, or you make iPSC from that. So it's your personalized stem cell, and you can use it for whatever differentiated cell you need. And unlike a cord blood, which is limited in amount, you can now expand this virtually indefinitely, so you can use it for many, many uses, and you're not limited by volume or the size of the tissue that you've collected. The other extreme is what companies prefer, because now that's very simple. You just make one line, and that line can be used in all individuals, because you've now either matched it or you do immune suppression in some fashion to be able to do that, and then you're only manufacturing one line, right? And so that's called the allogenic model, and that's what companies have tried to do. And if you look at examples of that sort of thing, it's MSC companies which say we'll take one line, we're going to make millions of products from that one line, and we're going to treat every patient who needs MSC from this one line, but it's not matched in any immune way. And there are many indications you could do that. And then there's the hybrid model, which I call the bone marrow transplant kind of model, where you have HLA matched cells. And then individuals come and you give one line to all the people who carry the corresponding HLA phenotype. So with iPSC, you can theoretically imagine doing all three things, right? You could take one iPSC line, and if it was really perfect, then you could use it in an allogenic model. You could make a few lines, but they were HLA matched because you use haplonull or some other strategy to be able to get a line which matched many people. And then you could go with one-to-one therapy. Okay. So the question then is, if that's great, and you can make iPSC, why am I talking about iPSC in a cord blood banking sort of session? And there's one important link between these two cells that I want to mention here. And so that's going to be in the last few slides that I'm going to go through, right? So one very simple thing is that the cord blood banks have experience in collecting tissue. And they know how to store it, and they know how to track it, and they know how to be able to move this sort of thing forward in terms of following the regulated guidelines. And because cord blood banks, the public banks, are actually licensed, you can now collect in a clinical fashion which would be approved by the FDA. So for people who are trying to do iPSC work, this becomes a shortcut because all of the hard work has already been done by the cord blood banks. So that's sort of one very simple reason why you might want to consider cord blood banks being involved with iPSC work. There's a second important reason for this, and I'm going to just run through the remaining slides and being able to do that really quickly to be able to do that, is it turns out that cord blood cells are the earliest cells that are present, so they're very naive, and they seem to be very efficient in being transformed into iPS cells. And a lot of people have shown that that can be done, and it can be done with very small amounts of tissues. So that was sort of second piece. A third piece, which is really important to keep in mind, is what happens to your body and when you're exposed to the environment. And all dividing cells collect mutations, and this is because you're exposed to the sun, maybe you're exposed to environmental toxins, etc. So all of these cause accumulated mutations which survive when you take an adult cell and make an iPS cell, right? So now you've got a possibility of having a pristine cell which can be harvested in relatively large numbers, and it's highly efficient in being transformed into an iPSC cell, and you have it collected and stored by people who have expertise under the appropriate regulations for being able to do it. So it seems like a reasonable possibility that this might be a way to go as a business model for making iPS cells. Okay. So we decided that we would try and see whether this would actually be possible, right? So it's all okay to talk about this in theory and say that it can be done with high efficiency, etc. And now ask ourselves, can this actually be done? And you know, I can't stand up there and say that my lab did it first or did it best at all. Okay, this was done by several different groups independently, and I'm not going to cite all of the people who did it, but several groups independently showed that we can take cord blood and we can take a really small amount of cord blood and we can take that and convert it into iPSC, and we can take the stored cord blood from a bank, right? So we, in fact, collected samples from the New York Blood Bank, we collected from the Carolinas Blood Bank, we've even talked with CBR about collecting samples from them to show that a cord blood sample which has been stored for as long as 6 years or 8 years can still be used to make iPSC. Okay. So that was sort of one very important piece. The second important piece was to figure out how much of the sample do we really need. And I won't go through the whole details about processes, and again, I want to emphasize that different groups have shown that this can be done. And one group which showed this, the smallest amount, was a group in Singapore, which said that we can do it from a single blood pinprick, so you can imagine the number of cells that are needed. But in our lab at New York Stem Cell Foundation, we were able to show that you can do this very easily and routinely in an automated way with 300,000 cells. You know, and that's less than 1 ml of a cord sample that you can use. So we actually took the tail and we cut that out, and we took that sample and we divided it into 3 and asked, can we take one of those samples and use it so that we had the other 2 as a backup to make iPS lines, and we could make good quality iPS lines. And that has been replicated and was done by several other groups as well. So on a practical level, we knew that blood that had been stored could be used, and we could make good quality iPS lines from it, and that there was a methodology that could be used to do it. So there's a third step in doing all of this, and that is the following. If I can make iPSC, can I make them in a clinically compliant way? right? That's the sort of $64,000 question here, right, in being able to do it. So that's what we checked. So there are several methods of making iPSC which have been described, which are clinically compliant. My group had been working with a CMO called Lonza to develop a clinically compliant way of making iPSC. So we went to them and said, we want to test whether we can take cord blood samples, use a clinically compliant, CGMP compliant method to make an iPSC line. And the answer then was, yes, they can do it. And that's, we've checked that, we've done all the manufacturing runs in a CGMP environment, and we think we have confirmed that that can actually be done. So that became the next really important question, right? We can do it, it can be done in a CGMP environment with materials and so on which have been licensed. So now you can imagine a path, right? You have a path where you collect the sample in an appropriate way, it's collected under FDA regulations, you've stored it in an appropriate way, there's expertise and database requirements for being able to track the cells and the sample, and you can keep it for many years, and then you can go back and make an iPSC line, and you can make it in a CGMP compliant method, right? So that was, on a practical level, also possible. So that was exciting. So then the next question when you get all of this is asked by people is, how expensive is this, right? Is this more expensive than any other method to be able to do it? And that's where we were lucky. Everything is expensive when you're doing clinical work, so don't get me wrong. I'm not trying to say that this is cheap. But what became very clear was that doing it from cord blood was, if anything, less expensive than doing it from other tissue sources. And so you had a quality advantage and you had an expense advantage because you had stored blood that could be processed and you could take a really small amount, and the efficiency of the process was quite high. So that led us to this thing about saying, boy, this does seem actually a practical level issue, and that we could actually get this done. And this would be one of the databases where we have HLA typing, so we could even get the haplo-null individuals so that we could make a bank of iPSC lines. So I'm going to skip this slide and go to this last slide here and then say, so then after I did all of this, when we talked about all of this with different groups, they said, so what's the real business model? You know, if we make hundreds and millions of lines for everybody before we know that the FDA is going to approve it, then it might be really expensive, because even though it's become cheaper and you can do it cheaply, making an iPSC line is not cheap, right? On average, the cost for us is about $10,000 if you're making it outside a CGMP environment. And the rule of thumb is that if you're making it in a CGMP environment, even though the processes are known, it's going to be roughly 10 times more, right? So $100,000 per person, you're not going to spend that up front, right, in being able to do it. So is there a business model being able to sort of consider making iPSC using cord blood as a starting sample? And so that's sort of my last slide here. So you can imagine that you don't make the iPSC unless people want it, right? You only store the samples, right? You store a sample in the right way so that it's under approved consent, and you store that. And then the cord blood bank can then either decide to make the iPSC itself, or it can partner with somebody who has the expertise to make iPSC and then be able to distribute that. You can also just say that in addition to banking the tissue sample, they can bank and store and distribute the already made iPSC, which might be made by some other group who spends the money to be able to do that. And they could also use their own expertise to be able to take the differentiated cells, which is what's the actual therapy, and become the shipping and distribution and differentiated cell partner, because they would have both the patient information and they would have shipping and contact information to be able to do that, and they would have validated processes to be able to do that. So I'm going to end here and let you hear from the other speakers as well. Thank you. So the last slide that I'm going to show you here in being able to do this is, and I'm going to leave it here just for a minute and just say, these are the questions for banking, right, that you have to worry about, even though I've told you that there's a theoretical path to being able to do it. You want to really decide which donor, on a practical level, which donor to prioritize, which sample to bank. Should I bank fibroblasts and blood or both? Do I want to do all three kinds of banking? Do I want to do one kind of banking? What should I do for the existing samples which have not HLA typed? You know, what would be the HLA typing strategy? Do I want to do high resolution? Do I want to do low resolution? How do I maintain uniformity across banks? Because it's not going to be just one bank, right, in terms of being able to do it. And then how do I obtain iPSC? Do I go with this Lonza method that I just told you about, or should I go with somebody else? Should it be standardized, or I don't need to have it standardized? That's all going to be important. And then, as you know, you can't get approval from the regulators for any of this piece unless you actually have an indication and you file an IND. So maybe there should be some decision on which should be the first disease as a showcase for what can be done. And then you also have to identify a partner who will manufacture the differentiated cells. So there are several different things that we need to consider, and I'll leave you with those thoughts to mull over when you want to ask questions. Thank you.
Our next panelist is Dr. Charles Cox. He's a professor of pediatric surgery and the George and Cynthia Mitchell Distinguished Chair in Neuroscience. He directs the pediatric trauma program at the University of Texas Houston Children's Memorial Hermann Hospital. He also directs the Pediatric Surgical Translational Laboratories and Pediatric Program in Regenerative Medicine at UT Houston. This program is a multidisciplinary effort that addresses problems that originate with the traumatic injury and consequences of resuscitation and critical care. The program focuses on stem cells for traumatic brain injury and related neurologic injuries. Dr. Cox, as part of this program, recently completed the first acute autologous cell therapy treatment for traumatic brain injury in children, and since that time he has been awarded several patents and many more INDs. Dr. Cox is a native Texan, and he received his undergraduate degree from the University of Texas at Austin. Then he graduated from University of Texas Medical Branch, then completed a surgical residency at the University of Texas Medical School at Houston. He did numerous further postgraduate fellowships in pediatric surgery, in critical care, and trauma. He's certified by the American Board of Surgery in surgery with specific qualifications in pediatric surgery and surgical critical care. He also served in Afghanistan in the 82nd Airborne in the 909th Forward Surgical Team in 2002. Dr. Cox has served on the scientific study sections and review groups for the National Institutes of Health, California Institute of Regenerative Medicine, among others. He needs to catch up to Dr. Rao. He's only published about, 150 scientific publications, 20 book chapters, and editor and text of a text entitled Progenitor Cell Therapy for Neurologic Injury. Thanks. We look forward to your presentation, Dr. Cox.
So as Heather said, I'm a pediatric surgeon and just down I-10 in Houston. And so I come at this problem from that perspective. Before we get started, there's the usual disclosures. We have funding from your tax dollars. We do sponsored research with different industries. There are advocacy groups who fund our work as well. And there's some very generous philanthropists in the Houston area who underwrite our work as well, for which we could not do this. So the purpose and what I want to try and communicate in the next 20 minutes is some proof of concept data in early phase clinical trials of neurological injury. And when I say that, most of this is going to be in traumatic brain injury, but I'm going to try and convince you that there's some correlates in other chronic injuries. To show you some preclinical and early clinical rationale for cell therapy in those chronic neurological injuries. And really, I want you to just keep in the back of your mind as we go forward of what we think the targets are in cell-based therapies, meaning specifically microglial activation or neuroinflammation and preservation of white matter fiber tracts. And then how that relates to cord blood treatment of neurological injury. So again, keep in the back of your mind what those clinical targets are, or those biological targets are that have clinical correlates. And specifically, we are focusing on white matter preservation and neuroinflammation. And those things, really, the knowledge of those things drives your cell choice. It drives the dosing regimen, meaning how much you give, when you give it, how you give it. And then the whole logistics train that follows with that, i.e. auto versus allo paradigms. So keep those things in the back of your mind, and I'm going to show you some data from our first clinical trials. So back in 2009, 2010, we completed our first phase 1 clinical trial in pediatric patients with severe traumatic brain injury. What we mean by that, by severe traumatic brain injury, for those of you who aren't in the field, that means that you're in a coma at that point in time of entry into this study. It's not a football concussion with a transient loss of consciousness, but you're in a coma. And we did a single-dose bone marrow mononuclear cell fraction study looking at safety. We've subsequently done a phase 1/2A dose escalation study in adults with 25 patients with a concomitant control group that was unblinded, but we did a dose escalation of 6, 9, and 12 million cells per kilo intravenously at 24 to 48 hours post-injury. And that has completed enrollment. And so back on the pediatric trial, one of the secondary endpoints was an imaging endpoint, and it was in global volumetric preservation of white matter and gray matter using a DT MRI technique. And this was published in Neurosurgery in 2009. And what you see is a study, a DT MRI that was done within the first month post-injury, and then one that was done at 6 months post-injury. And what you see is there's no difference in gray matter GM and white matter WM volumetrics on either of those. And so you kind of say, well, so what? Well, the so what is that in our same institution in age-matched and severity-matched patients, there's about an 8 to 10% difference in gray white matter volumetric loss in patients at the same time point post-injury. And that's been shown in a number of places across the literature, both in adults and in pediatric patients, that 6 months to a year post-severe injury, you lose between 8 and 15% of gray matter and white matter volumetrics, and it's replaced by cerebrospinal fluid. So you have ongoing cerebral atrophy. So to show you some data from the adult study, we did some more sophisticated DT MRI imaging that also focused on corpus callosal fiber tract mapping on top of the white matter volumetrics. So the corpus callosal, for those of you who aren't in neuroscience, is a strip of tissue that connects the two hemispheres in your brain, and all of those fiber tracts provide the associations between the cortices, and really injury to the corpus callosum is what causes decrements in your executive functioning and information processing, et cetera. And so we had some very interesting data from these studies. So this is the type of map that's generated. These are all individual fiber tracts. This is my brain, actually. And what you can see is the front to back, and then looking down on it from above. So you can see, and an FA, fractional anisotropy, is a measure of how well the cells are, those tracts are myelinated. And the more orange they are, the better they are in terms of conducting information. And so this is the first important thing that I can show you, and that is that if you look at gray matter volume, white matter volume, no treatment in the blue bars, there's about combined a 6 to 7% volumetric loss of total gray matter, white matter volume. I'm not talking about the corpus callosum now, but just kind of like that first slide I showed you. In the therapy group, when you pool both the 6 and 9 million cells per kilo doses dose groups, you can see that that's blunted, and it's about a 1.5% total volumetric loss. And it seems to be dose dependent. You see how you get up to the higher dose at 9 million per kilo. We haven't the follow-up on the 12 million per kilo patients yet. They're still accruing. But what you see is that instead of this 6% volumetric loss, you have less than 1% volumetric loss in this higher dose. And what's encouraging is it seems to be dose dependent. So that's an interesting point. And now what I want to show you is a couple of individual patients who, this is an untreated patient, 1 month and 6 months, the same two type views post-injury. You can see at 1 month there's already volumetric loss, and you remember my brain. But look how there's even further dropout at 6 months, and there's this central dropout at the 6 month time point in the corpus callosum, as opposed to a treated patient where there's volumetric preservation. There's still ongoing loss, you can see here and here, but it's not nearly as severe in the treated patient. Now with the corpus callosal fiber tract mapping, the imagery can count each one of these individual fibers, okay? And it can also quantify globally what's called the FA, the fractional anisotropy, and the MD, mean diffusivity. So that's how well the cell is functioning in terms of maintaining its ionic barrier, and that's a prerequisite for function. So this is probably the most important slide from that piece of information. So if you take those fiber tracts and you count all of them, now that's not measuring the FA or the mean diffusivity, but it's the presence or their absence Okay? In the no treatment group in the blue bar, there's a 10% loss, about what the literature would suggest we see. We were seeing the same thing. Low dose, 6% increase. High dose or medium dose, 13% increase. The high dose, we're still collecting that information. But again, reassuring that it's dose dependent, reassuring that we saw what was previously reported in the literature. And the reason that this is important, white matter volumetric preservation, corpus callosal fiber tract preservation, is those two things correlate very, very strongly with clinical outcomes. Okay? And so let me show you some of those clinical outcomes with which these correlate. This is the fractional anisotropy I was talking to you about, that the more orange the fiber tract maps are. And then these are all neuropsychological outcome and functional outcome measures. Glasgow Outcome Score is a global functional measure that measures everything from death to normal and in between. And these are regions of the corpus callosum, and this is the whole corpus callosum. It's FA, so it's global degree of myelination. The yellow highlights are those that are statistically significant. But what you see is that for the whole corpus callosum, for a whole bunch of these neuropsychological outcome tests that measure your degree of executive functioning, your ability to do tasks that make up your day, that there's a statistical improvement in those tasks that correlate with the degree of fractional anisotropy or volumetric preservation and health of those corpus callosal structures. Is that a definite causative link? No, it's not. It's a correlation. But because it's both dose dependent and it shows that splay that is consistent with the previous literature, it's very promising in our mind. How do we think that this works? Remember I was talking about neuroinflammation in the microglia. We think that the infusion of these cells alters the systemic inflammatory response to injury. These are dendrograms that don't press me on the math here, but it measures the relative similarities and differences of groups statistically. And this is TNF alpha, IL-1 beta, pro-inflammatory cytokines. These are both reduced with cell-based infusion. Anti-inflammatory cytokines, IL-4 and IL-10, are increased. No change in IL-6 or interferon gamma. So what we see is at this 48 to 72 hour time point post-treatment, there's an alteration in the systemic inflammatory response in the injury, and it's across multiple cytokines. So it downregulates the inflammatory response, upregulates the anti-inflammatory response. The reason that's important is those cytokines act upon the microglia within the brain, which drive some of that white matter preservation. Whoops. So what about neonatology and umbilical cord blood? What you can see here, this is lifted from Dr. Zhu, Cytotherapy 2014. It's a great diagram. And it shows all of the different diseases in neonates that are potential therapeutic targets, but I want to concentrate on this one in the brain since that's what we're interested in. Intraventricular hemorrhage and PVL, or periventricular leukomalacia, those are two drivers of cerebral palsy. So that's a chronic neurological injury, right, if we're seeing those patients later on. Why might that be, how might that work? I was skeptical about that for a long time. There's a great work that came out of a group in Germany back in 2006 that has a rodent model of cerebral palsy, ischemia, and hypoxia, so the two of the prerequisites. And what they showed is that when they used human umbilical cord blood transplantation, it decreased the spasticity as measured by the foot-paw width and the degree in which they could step. And so these are little paw pad prints up here. And if you've seen children with cerebral palsy, you know that they have an upper motor neuron disease. They have a spasticity, so they are unable to extend their limbs. And so same way with these rodents, they're unable to extend their paw, and they can measure that. And you can see that when you have the injury and you have cell transplantation, it looks like the control group, and there's no difference, both in the degree in which they step and the degree of spasticity. Importantly, none of this work with any of these mononuclear cells is speaking towards transdifferentiation of these cells into neurons, astrocytes, or other types of cells, okay? And they didn't show that either. They showed that there was migration, the green labeled cells, the red are as a neuronal stain. Transplantation, they did have migration to this area of ischemia, but there was never any evidence of transdifferentiation. So this issue of brain ischemia, the target is this white matter. So this is partly why I got interested in the cerebral palsy thing, is the injury is a white matter fiber tract injury. That happens to be where we were seeing some improvement on the traumatic brain injury patients, right? And that's what drives the clinical syndrome. This is the brain, this is the lateral ventricle of the brain, and this, that's where it's called periventricular. That's the area of abnormal circulation that's most affected with hypoxia and ischemia. And we were kind of late to the game in this cerebral palsy thing, but this is a paper that really started to get me thinking about that as a possibility. So this was published in the Annals of Neurology back in 2011. It's from a group in London. And this is a PET CT scan. Okay, so, and they have a label, PK11195, which when infused binds to activated microglia. Okay, and so you can image that. These are PET CT images of patients' brains. You know what? These numbers down here are the number of months post their traumatic injury, which still showed activation. That's all these different non-blue colors in the thalamic regions of the brain and in the periventricular regions of the brain. So there's chronic inflammation way out past that initial TBI, which gave us pause. And what's the important thing is that the degree of that neuroinflammatory response inversely correlates with your level of consciousness. So the more ongoing chronic neuroinflammation there is, the more depressed your level of consciousness, specifically in the thalamus, which isn't surprising for the neuroscience crowd. Other data came out in animal models, so we always debate what 1 year is. It translates in the life of a rat to a life of a human, but it's a fair amount of time. It is definitely chronic, that there's still activated microglia on histologic sections even a year out. That's all these black stains. 1 week, obviously more, but 52 weeks out, there's still this ongoing chronic microglial activation. And the more chronic microglial activation you have, the more white matter volumetric loss you have. So you see where I'm going with this story, that we feel that we have an ability to alter some of those readouts. And I haven't shown you our animal data on microglial activation, but we have a lot of preclinical work to support that. To where we now have a clinical trial that is ongoing for cerebral palsy in children age 2 to 7, I think I have that right. That is a blinded, randomized crossover design. If you have umbilical cord, you go into the umbilical cord blood side, you either treat it or control, bone marrow, treated or control, and then you go out for 1 month, you get those same volumetric DT MRI imaging. And the beauty of this is it's a paired design, right? We test you before and after. So we were comparing an individual's response before and after. And if you're not treated, we break the code, you can cross over and get treated from here out, right? So everyone has an option to be treated, but we don't know who that is until we break the blinding at 1 year. We've done 10 of the 15 bone marrow arm. We have a big line for those, and we've got 4 of the 15 of the autologous umbilical cord blood. To summarize the published cord blood clinical trials in CP, these are the ones that we can find that are published, and it's a little bit of a mishmash. This one has EPO preconditioning and shows improvement in their gross motor functional outcome, and that correlates with the fractional anisotropy. So very similar data to what we showed in the TBI, and their functional outcomes on all of this preconditioned umbilical cord blood is greater than controls. It's hard to know, teasing out the EPO effect versus the umbilical cord blood effect. It wasn't a perfect clinical trial design. This shows a improvement in 25% of the patients, but there's an age break point of benefit. Once you're up after 3 years of age, there doesn't seem to be a benefit. This is shows an anti-inflammatory cytokine response similar to what we'd seen in TBI. It was very small numbers, and you can't really make anything out of the functional outcomes. Here's what's ongoing on clinicaltrials.gov. Joanne Kurtzberg has the largest trial going. She's been a leader in this field. My understanding, she stopped this trial at 70 patients. I know that there were some secondary readouts that were positive, and I don't know what the results are, but she has some very sophisticated interactive imaging that is going to be exciting to see. The group in South Korea, Dr. Min, has a host of studies, and it's hard to tell precisely on clinicaltrials.gov which one overlaps with which one, but they have an ongoing study. There's ours. Georgia Regents University has one that's ongoing. No imaging readout there. And then in Mexico, there's one that's not a typical clinical trial design. Broad overview, but I hope I can convince you that at least there's some strong preclinical and early phase clinical data that's autologous cellular therapy for neurological injury is a reasonable path moving forward. And we think that those targets have to do with inflammation, with principally the end effector cell being the microglia, with the downstream result being white matter preservation. We think that imaging with a microglial activation is becoming a biomarker that is relevant for treatment efficacy, and that umbilical cord blood now is representing a potent therapeutic option and these phase 2A and B clinical trials are ongoing. I'm over time.
All right, our final presentation will be by Frances Verter, and she's the founder and director of Parents Guide to Cord Blood Foundation, which is a 501c3 charity that serves dual roles as cord blood information resource for both parents and professionals. Dr. Verter started her career as a research scientist, earning a BS degree in physics from Brooklyn College and a PhD in astrophysics from Princeton. She embarked on a career studying the formation of new stars in the dense gas clouds of the galaxies, eventually joining the staff at NASA's Goddard Space Flight Center. At this point in her career, she became a living example of the saying that says, life is what happens when you're busy making other plans, immortalized by John Lennon in the song Beautiful Boy. Dr. Verter's first child, Shay, was born with a rare cancer. Their quest to find a cure forced Dr. Verter to spend more of her time with her daughter than at NASA, and they really became the first stem cell tourists before the phrase was even invented. They traveled within the U.S. so that Shay could receive the optimum treatment protocol for her case, but unfortunately Shay passed away. Dr. Verter started a second career at NASA doing computer programming for climate models, and then she gave birth subsequently to 2 other daughters. During this phase, she launched a Parents Guide to Cord Blood website in 1998 in memory of her daughter. Originally, the website was only intended to be a hobby, but gradually it's taken over Dr. Verter's life. The Parents Guide to Cord Blood became incorporated as a foundation in 2007 and has been growing steadily ever since. It's a recognized consumer resource helping families to learn about both cord blood donation and family banking. Professionals also rely on the foundation's unique database of cord blood banks around the world. Dr. Verter fervently hopes this third career is her final reincarnation, and based on what we've been hearing about cord blood stem cells, it's a ripe field, so I think it will provide her a lot of satisfaction.
Since I'm all about parent education, I'm going to try to stay grounded and actually stay on the ground. So let's see, how do we forward it? Just go this way. So the first part of my talk, I'll tell you a little about my foundation, if there's anybody here who's not familiar with it, and then the second part I'll talk about cord blood clinical trials. And I'm going to try to be quick so we can all, go to lunch on time and ask our questions and things like that, which I know are crucially important. I wanted to throw up the homepage of the website. That's actually the homepage for searching if you want to donate cord blood. One of the most important things about the Parents Guide is that we try to stay down the middle and not tell parents what to do. We try to teach them both about donating cord blood for the sake of society and helping patients who need transplants. And we also educate parents who want to bank privately for their family on where to find a bank and how to pick a bank. but we stay neutral. We don't tell parents what to do, and it very often puts me in a difficult situation when parents want me to tell them what to do, and I try to just say, here are some good choices and pick one. So that's our primary focus, is to educate parents with accurate and balanced information. Our secondary piece of our mission statement is to do cord blood statistics, and I'll be talking about that a bit more later on in my talk, to do statistical studies of the field. So how many people in this room have personally been interested in banking cord blood or donating cord blood for themselves or a family member? You know, how many people here are actually consumers? Okay, so pretty good show of hands if people had, you know, a reason to look at the Parent's Guide for consumer information. And how many of you are actually professionals who work with cord blood? I know you do, in the cord blood industry space. Okay, so quite a few. I think some of you are keeping your hands down, but really should be raising them. Okay. I know you guys. So our website serves both audiences. In any given month, I'm going to say roughly 75% of our readers are parents who are looking for information, and they're newcomers to the website. When parents are having a baby, it's like consumers where their car broke down and they suddenly need a car. All of a sudden, it's like, I have to read about cars. And they show up at the website and they read all about cord blood and they make a decision, and then they disappear and they don't come back until a few years later. And I've had parents say, oh yes, I used your website for my last baby and now I'm back. So they're like a revolving readership. And the other, like 1/4 roughly of our monthly readers are people who are in the industry who use our website as a resource and are constantly coming back to find a scoop on their competitors or look for clients. So just to review things that we do, we're a nonprofit. You should support our work, make a donation. It would also help me if you give me a 5 on your speaker review. That way I get invited to give more talks. So we have maps of cord blood banks around the world, and you can search in any given country, find the list of banks, find a grid of their prices, their accreditations, pick a bank. Because there are other organizations that will tell you about public banks. You can always go to World Marrow Donor Association and buy their annual report. Purpose of our website is to help everybody who's looking for the family bank information. We do have the donation locations within the US and Canada, and that's a searchable grid down to the state level. You can find the nearest hospital to you that collects cord blood donations. We are rolling out some new initiatives for both parents and the industry. For parents, we have launched a mobile version of the website just within the past couple of months, and it still has some glitches, but if you have phones, you can look up the URL that was on the previous screen, parentsguidecordblood.org. Try looking it up on your phone and see how the website looks and send me complaints about things that need to be fixed because it's new. But it's important because you know, when you're doing consumer education, you have to meet consumers where they are. People who are having babies all carry phones. They tend to look things up on their phones. They want to do this and figure out which bank they're going to click on and call. So it's very important to have a mobile version when you're doing consumer outreach. For the industry, our new initiative that's coming out in early 2015 is going to be a cord blood industry report where we will have confidential information about cord blood banks around the world that is not published on the website. It will be the very first industry report in this space that's actually based on interviews of individual companies and is not just compiled from public databases. And we'll have things about processing systems the banks use. We will have the inventory only in aggregates because we promised everybody CBUS when they told us their inventory. And we will also have statistics on how cord blood is being used by different banks in different parts of the world, what therapies are they releasing, but that's also in aggregates because we promised everyone confidentiality. So that's what we do, and we've been recognized. We have formed partnerships with the main accrediting agencies. FACT, NetCord, and AABB are the 2 premier accrediting agencies for cord blood banks. And we do also have other services. Let's see. We have brochures in multiple languages for multiple countries. In the United States, we have English and Spanish. And they're free. You can tell your OB-GYN that they should carry educational brochures, that they should put one in the packet for every pregnant mother, and we will get them free brochures. 6 states have accepted the Parent's Guide as their educational resource used by their Department of Health. Thank you, Brian, for giving me the update. Oh, and we also have a newsletter on the website that comes out monthly. That's free. And just basically, that's all about the foundation. So let's move on. Let's talk about cord blood applications, because I know we all want to ask questions and go to lunch. Part 2 of the talk. So currently, there's a standard list of 80 diseases treated with cord blood, and CBR has this really cool little handout lists all 80 that you can get a copy of. Particularly if you have a medical practice, it's kind of useful to hand out for education purposes. But the important thing about the 80 diseases, and it's roughly 80, but people round off, these are all diseases where the FDA has approved the use of cord blood for a cord blood transplant. So we've got a range of disorders here. We've got cancers, obviously, leukemias, lymphomas. We also have blood disorders that can kill you without being cancer. Severe aplastic anemia, severe sickle cell. These things can be fatal and can be cured with a cord blood transplant. Immune disorders, too. Many of these are extremely rare. You know, even though there's a list of 80 diseases, some of these diseases, there are only like 3 children in the US that have this disease at any given time. Some of these are extremely rare. And then there's also the metabolic disorders that are now considered a standard treatment with cord blood. But all of these collectively, by the age of 20, the likelihood of having one of these treatments is only 1 in 1,667. Or in other words, 3 in 5,000. I mean, it's really not very likely to need cord blood for these traditional applications. What's becoming the more compelling reason for people to be interested in cord blood is the clinical trials. And so I'd like to talk a bit more about the clinical trials. Let's stay out of the way if anybody wants to take a picture of these trials. I've put together, well, on my database, my website, I have the current clinical trials with cord blood. Primarily, they are for young children to correct brain injuries that occurred near the time of birth. And this could be HIE, which is short for hypoxic ischemic encephalopathy, and it leads to cerebral palsy, or CP. There's also some new trials that started in the past couple of years to see if cord blood can help autism. And we also have a trial that is looking at acquired hearing loss, where there's a loss of hair that conducts hearing in the ear. The ones that are in bold purple bars are trials that are supported by Cord Blood Registry. They are supporting more FDA-approved trials in the US than any other private cord blood bank. Cerebral palsy has really been the big booming application for cord blood. There are many children who have reported dramatic recoveries in these trials. And as Dr. Cox mentioned, Dr. Kurtzberg has ended her trial early, which would imply that she has positive results to report. And we're all hoping that this is going to soon become a mainstream therapy. You can see that there are countries around the world that have started their own cerebral palsy trials. There's US, Mexico, Singapore, Korea, Japan, Romania, which is actually a hotbed of cord blood banking, and Australia. But you can see that the original trials for many of these disorders started in the US. So the origins of cord blood banking were in the US, and we are still the first to try some of these new applications with cord blood. I have been talking strictly about cord blood and not cord tissue cells. So I'm trying to keep this restricted to one topic. Okay. So to give you an idea of how likely you are to need cord blood for a clinical trial, as I had mentioned, the odds of needing cord blood for a transplant are really not very likely. I personally wouldn't lose sleep over it, but when you look at how many children have cerebral palsy or have autism, I'm sure that everybody knows like a neighbor or friend in their community that has this. 1 in 300 children who are in elementary school have cerebral palsy. And then the incidence of autism has been increasing in the United States. It is now up to 1 in 68. The CDC released that number in March. I mean, my best friend has a son with autism, and I wish this trial had come out when his son was young. I mean, this is something that I think probably touches most of us. Hearing loss, acquired hearing loss is really, really prevalent. That's a very new trial, but that's a 1 in 8 situation. I personally suspect that wearing earbuds is going to make it even worse. I mean, in my age, we played our music too loud with speakers, but now people are doing it directly into their ear. So the likelihood of needing cord blood for some type of application is really rising, and this is pushing parents to take, you know, cord blood banking a lot more seriously. I feel that the future is coming where cord blood banking will become mainstream, because we had started with the title of this whole, you know, panel is the future now? And I feel that future is coming. This is a graph of cord blood therapies released from just one bank, the largest private bank in the US, which is Cord Blood Registry. You can see that the rate at which they have been releasing therapies for children has been rising dramatically. And therapies for these various regenerative trials that they're involved in has become their predominant form of releasing cord blood. And they're to be commended for being involved with so many trials, because I track cord blood banks around the world, and I find that the rate at which banks are releasing inventory, releasing therapies does not automatically track when they have bigger inventory. I mean, I see banks with over 100,000 and their release rate is way lower than this. It's because they're not affiliated with cord blood trials. What really drives up release rate is being involved in trials, having relationships with doctors. That's what causes banks to have high release rates. There are some cord blood banks or private banks that have release rates comparable to public banks because they're heavily involved in trials. So I admit that I'm a little biased because I track cord blood usage, but I want to emphasize to both parents and professionals that it is the most likely to be used in the near future because of the ease of getting, you know, cord blood into the clinic. A lot of the problems with the FDA approval that so many other stem cell sources have, or so many stem cell preparations have, cord blood does not have, because you can get effects just using the stem cells themselves without having to expand them, manipulate them. You can meet FDA approval. You see that there are all these trials going on, like Dr. Cox's trials, and the other trials for cerebral palsy, autism, hearing loss. You know, it's easy to get cord blood at birth. You know, it's not invasive to harvest it. I also want to point on the fact public banks very often say that, oh, you know, there's rarely enough cord blood for clinical use. Well, that's only if you're talking about transplants. You know, if you want to transplant the cord blood, you need a big collection from a jumbo full-term baby in order to transplant an adult. But parents who are having just an average-sized baby will have plenty of cord blood for regenerative medicine. In fact, for multiple therapies, you could divide up your unit and use it a few times. You do not need a big collection for these clinical trials, and that's another important increase in accessibility of therapy using cord blood. You know, I think I've covered most of my points, and I have, you know, that wonderful fuzzy picture, and I guess now we'll take questions.
Thanks, Fran.
I kept us on schedule.
So before we open up the floor to questions, let's start a little discussion. I was going to throw it out to all of you, but maybe I'll just throw this one out to you, Dr. Cox, since you are in the lead seat there. What do you tell friends, family members, other physicians, expectant parents you run into about umbilical cord blood banking?
So, I mean, I advise them to do it. I tell them, you know, it's there are two issues, right? It's a cost-benefit discussion, and so for the vast majority of people who can afford it, I advise them to do it. They ask kind of really get at kind of what's the likelihood of use is the question, and I don't kind of get into those numbers like that of, well, it's 1 in 16,000 for oncologic, I mean 1 in 1,600 for oncologic, and this. I said, you know, there are people who will tell you it's asteroid insurance, right? And then there are people who tell you it's like having car insurance. It's probably somewhere in between. It's more like if you live on the Gulf Coast, flood insurance. There's a good chance that, you know, there will come an opportunity that you may need a unit, but it's not something that's, you know, it's somewhere in that range is how I explain it. And then I think most people can walk away with that. I tell them, you know, if you're trying to figure out if you're going to bank cord blood or pay the mortgage, I say pay the mortgage. But you know, if it's something that's that can be dialed into your finances, I say that it's worth doing.
I see nodding heads on the panel. Do either of you have anything to add to your counsel to patients?
So I think the one time when I really push the idea is if you've had the first child which has had some kind of hereditary disorder, because then the possibility of use and benefit on a personal level rises much more, given the numbers that you've pointed out. And I tell everyone that, you know, if you don't want to bank for yourself, you should consider donation to a public bank in any case, if that's available.
Fran, you operate an educational website. Do you still think there's a lot of misperceptions, misunderstanding about the consumer and general public about umbilical cord blood stem cells?
Well, it's interesting because the Parents Guide is international, and in the United States, I feel that consumers have become a lot more sophisticated over the past 10 years. And usually when people contact me, they already know what they want to do, and it's a question of when and how. And actually, the majority of my inquiries are parents who want to donate, because there are plenty of private companies that would be happy to give them information about private banking, but with donation, it's either me or the NMDP, and they have questions about maps, of where to go, and timing of whether they're too late to register. I find that my second biggest source of readers is India, and India is very similar to the United States in the 1990s, where the banks are pushing a great deal of misinformation that I have to correct. So it's a different ballpark depending on the nationality I'm dealing with. In the US, I really think it's important that we have programs for the high-risk moms. Any mothers who are in danger of having a premature baby really should bank cord blood privately, because a small baby will not have enough cord blood to donate, and they have 10 times or higher risk of the various conditions for which cord blood is in clinical trials. So I really think we should have more of those programs where at the hospitals where there are high-risk mothers, there should be education campaigns about cord blood banking.
Dr. Rao, in your presentation, you mentioned the data that supports that in a traditional transplant use, a related umbilical cord blood sample performs better than an unrelated, but still matched cord blood unit. Is that going to translate and be the same when we talk about regenerative medicine therapy, some of these applications that we've discussed this morning? Will your own or a related familial sample always perform better?
So I want to make sure that we are clear on this. It's not that it always performs functionally better, right? I mean, if you have a hereditary disorder and your sample has the same hereditary disorder, it's not going to perform functionally better. But for the immune system and rejection, it's very clear that your own cells are recognized as not being foreign, with much less rejection rates, even if it's a non-blood transplant, right? With the bone marrow transplants, you also have to worry about graft versus host, right? But even for tissue that's an immune privileged site, at least the animal data suggests that if you have your own cells for the animal, that they perform better in the long run when you're looking over years than if it's a mismatched cell. even in an immune privileged site. So I would assume that that's going to be the case for most regenerative medicine purposes.
Dr. Cox, in the clinical trial space, have you seen any barriers to conducting clinical trials with newborn stem cells? Is there anything else that needs to happen for newborn stem cells to reach their full potential?
I think that the barriers are a fewfold. One is for acute applications that requires a substantial logistical footprint to do that at typically more than one site or one or two big sites, and that's a significant capital investment. Then on the autologous side of things, it's having enough patients who have already stored cord blood, who have a condition for which the trial is active. So if you looked at, so for example, Joanne's trial, our trial, Georgia Regent's trial, or places that are using autologous therapies, all told, that's about 100 patients or so, maybe let's double it, 200 patients, right? Well, that's a barrier in terms of the autologous side, is that there's not enough autologous patient. Plenty of people with CP and plenty of kids with HIE and chronic neurological injury from trauma. But when I've overlapped that Venn diagram with, and I have cord blood storage, that becomes a really, really narrow number. And so I think that those are the two big barriers. So then you have to either have casting a wider net in terms of your autologous collection, or come up with some other model for capture.
Or basically allogeneic trials, using cord blood from a donor for brain injury.
Or allo trials, yeah.
We're going to take questions at the microphone. So Kyle, if you'd like to take the first question, and I'll ask the audience to please approach the microphone if you'd like to ask questions of the panelists.
So my question kind of contradicts what Dr. Cox just said, but I was thinking more along the lines of, are the cord blood banks prepared for, if this autism trial that CBR is sponsoring, which I think is awesome, by the way, and all the sponsorships that you guys do with cerebral palsy and whatnot, but if they get great results with the autism trial, you probably have 10,000 plus families that maybe have an autistic child in your inventory, I would guess. I mean, at the odds of 1 in 100, and you have 500,000 units. So is the industry really prepared for every one of those families would want to go through a cord blood regenerative medicine transplant to treat the autism? And I was just wondering if, to think ahead a little bit about the demand to use the banks. I mean, sending out 300 units in one a week is something, sending out 100 a week is totally different. Where are they going to go? Who's going to do the transplants, and what's that going to look like?
I don't know if you want to speak at it from the clinical trial perspective.
Yeah, well, so I mean, we, at least on the CP side, we're not, we don't see those numbers in the banks, or at least we don't capture those numbers in the bank for whatever reason. Autism may be another story, but I do think you do have a good point in terms of the logistics of release are a whole nother component to being a potential barrier. Yeah, I mean, it could be a big deal.
Dr. Rao, do you have some other comments?
Yeah, I just wanted to add one more thing from Dr. Cox's presentation, and that's, while you're absolutely right, there are going to be logistical issues. Remember what he said about the fact that generally most benefit in a lot of the trials has been before 3 years of age because of what we think of as the blood-brain barrier in terms of therapy and delivery. And so while we expect, even if the results are awesome, that we'll still have a more limited number of patients than all the people who are in the bank to consider at least as a first stage. So we'll have time. And the other piece to remember is that there are 30,000 bone marrow transplants that are done every year. So clinicians have that expertise of being able to do it, and they should certainly have the facilities and the infrastructure to do it. Now it's just for a different indication. So I don't think it will be a huge problem. And if NMDP could figure out the logistics, I'm sure the cord blood banks will be able to figure that out.
I agree very much with that, and for models that are being developed like the Serum Alpha Clinic model, where there'll be an existing coordinating centers who are used to doing these kinds of treatments as they're established. Next question at the microphone.
Thank you. It's really
it's not on.
Panel discussion.
It's not on.
It's not on. So how can I fix this? Is it here?
Speak louder, I guess.
Okay. Well, I'm Xia Hongfan, medical officer at the FDA Center for Biologics Evaluation and Research. Thank you so much for the excellent panel presentation and discussion. I do have comments for Dr. Rao and the questions for Dr. Cox and Dr. Verter. I missed Heather's presentation, so you are excused, but I'm sorry. Now, so first of all, regarding Dr. Rao, I really appreciate your presentation and a lot of information about what the FDA is doing, the regulations, and I appreciate that your understanding of the work, and it's excellent. So just one comment about, I want to also invite everybody or make this plea, sponsor, investigator, that we do want to be your alliance. Don't be afraid of contacting the FDA. You are always welcome with your questions and issues at any stage of your development, if you're just thinking. So, and FDA, we give you free advice, pre-IND, IND, only until you, at the end, for example, you conduct a phase 3 study, you are ready to market your product. Then we will evaluate the evidence for the efficacy and the safety for the process of the marketing approval. Only at that point, we may or we will require the user fee payment. However, there are also waivers for some of the product. It depends. I will not come to the details. There are waivers, so you maybe don't have to pay any fee for even for the final stage. Our mission is to facilitate the development and advancement of the effective and safe drug and biologics.
Thank you for that. Thank you for that comment. As a bank that I've been through now 6 IND review processes with the FDA, and it's been very helpful and very informative in the process. So thank you for that comment.
Oh, great. I'm glad you're aware of that. The second part is...
we do only have just a few more minutes and other questions behind you. I'm so sorry.
Thank you. Very interesting presentation. This is kind of a question from a layperson. So I've got 5 grandchildren, 7, 5, 5, and 7, 5, 5, 2, and 1. All we saved their cord blood with your firm. So how do you? And I don't know it because their parents did it. But what is your typical division of units? In Dr. Rao's presentation, I think you described, or one of you described, that you can't unfreeze it and refreeze it. So, and so most of the people that collect, I think, are collecting for the health or future use of the children. But there also appears to be uses for the parents. My daughter's got Crohn's. So you wonder, could she use some of it for this and use the iPSCs for that? And if so, possibly it's a little selfish to take some of her children's cord blood to do that, unless you can take some of those units and subdivide them while they're still frozen and use them. So,
yeah, that's a great question. So there are variables, but the standard array of storing an umbilical cord blood unit is in a freezer bag that's about the size of a deck of cards. And that's divided into 2 compartments, a minor compartment and a major compartment. About 80% of the cord blood content is in that major compartment, about 20% in that minor compartment. And as Dr. Rao alluded to, there's a couple of segments that we call pigtails attached to that Mostly right now, those are just used for confirmatory testing, some regulatory testing we need to complete before a unit is released for use. In a traditional transplant use, blood disorders, cancers, genetic disease, transplant physicians want every last stem cell available to treat that patient. In some of these new emerging regenerative medicine therapies, the cell dose required to treat a patient is really unknown right now, but certainly much lower than when somebody's going through chemo or radiation for a transplant medicine procedure. So in some of our own trials, we've been able to release just the minor compartment or just the major compartment because it's sufficient for the treatment, and we reserve some of those umbilical cord blood stem cells for the family. We've looked at ways to divide the cord blood unit multiple times and store it in smaller aliquots, but sort of we have to put some, for lack of a better term, antifreeze in with the cord blood, and that doesn't mix as well when you divide it in smaller and smaller compartments, so that hasn't been perfected yet. But certainly iPS cell generation can be achieved through a small amount of cord blood stem cells, so we could take a small amount of blood or tissue to create an iPS line, and stem cell expansion, which obviously we don't have time to cover today, and I don't think is covered in any of the other talks, is a possibility in the future where we could take a portion of the cord blood and replicate it for multiple future uses for a single individual or multiple family members. Other questions? Yeah, thank you for staying with us through lunch. If you have questions, we will be happy to answer them.
You shouldn't have told them that lunch had started.
My name is Shangqing Guo. I'm assistant professor at Yale University Stem Cell Center. I'm also a customer of yours, CBR. So in the lab, we use blood progenitors or stem cells to make them into becoming other cell types. And because of the ease of doing engineering, we just have so much more understanding mechanistically and so much better control in pushing them into different things. Now what we'd like to do is to test whether our knowledge can be similarly applied to cord blood stem cells and make them into different therapeutic cell types. However, the big problem here is that because we're jumping between species from mouse to human, and the funding agencies are very suspicious. And the first question, or I got repeatedly asked about this, show us the human data that you can actually do similar things with human cells. And so this is where I got stuck. So we're trying to bring our knowledge obtained from mouse cells into the human systems, but so far, we don't have sufficient support. I'm just throwing the question out there. How do we bridge this gap?
So that's been the one huge plus, and why we pushed with the cord blood banks is to make the research grade samples available. And when I worked at the NIH, we actually had a program where we would collect just for research cord blood samples which were too small for actual therapeutic use, and we would supply people with cells from cord blood for that kind of research use, because there are these differences between mouse and human, and there's a big effort to make what are called humanized mice so that you can test human cells in a mouse model, be able to get those sorts of results. And so that's been a big activity, and we think it's a very important piece to get the kind of discoveries which would be translatable. So. Thank you.
Yeah, Fernando Pitosi from Buenos Aires, from Argentina. I have questions and a comment for each of the panelists. So, Dr. Rao, if you have to compare the three models that you have presented, one-to-one, one-to-many, and many-to-many, and you have to bet in the future, will there be space for the three models to coexist, or will you think one of these models will prevail as a method of transplantation and use of these cells?
So I think the one-to-many, which is the allogenic model for a lot of these indications, is probably not viable, even though it's cost effective because of all of these immune issues. So I think the other two models are likely to both coexist rather than not exist at all. It's unlikely it will be completely personalized because of the expense and the scale. You know, maintaining millions of lines is going to be very difficult. So some hybrid model will probably be the most likely.
Okay. And for Dr. Cox, do you think, or do you have evidence that all umbilical cord cell preparation will be effective in the treatment, or do you think there's going to be some standardization needed for each preparation to be regarded as a product so that you see these immunosuppressive effects on the patients?
I'm not sure I understood the question.
If you have any, for example, control of what are the characteristics of the cells you are injecting to predict the effect of the immunosuppressive effect you see.
Yes, and so in a host of different, I'll just call them, for lack of a better word, cousin cells, MSCs, other bone marrow derived cells, mononuclear cells, etc. We have in our preclinical injury models, a readout of blood-brain barrier permeability as a index of in vivo inflammatory response that's clinically relevant. And some cells work and some cells don't. And when we take those cell lines back and do a potency assay panel or matrix, if you will, what we found is that PGE2 secretion, TNF alpha suppression, IDO expression, and those, if you look at those kind of in total, those correlate, those readouts in a mixed lymphocyte type reaction, not precisely, but that format of a mixed lymphocyte reaction really predicts the in vivo readout that we think is clinically important for our moving forward on trials. So yes. Okay,
so you can select the preparation before infusing them. Okay, thank you. And just a comment, there has been an approval from the National Regulatory Agency in Argentina for a clinical trial using umbilical cord stem cells for hypoxic ischemia in newborns a month ago. It's not on clinicaltrials.gov, but it will be there.
You need to send me information so I can get it into my database.
Ken, thanks for being the last question at the microphone. If you have additional questions for the speakers, we'll be available for the podium following.
Okay, thanks, Heather. Excellent session, actually. It's great to see cord blood being supported clinically now. This question is for Dr. Cox. It was very interesting on your anti-inflammatory data. And were the measurements of the cytokines or the anti-inflammatory proteins, were those done in peripheral blood or were those local from tissue extracts?
Those are peripheral blood samples from our adult phase 1/2A clinical trial. We have a minority of the patients we have CSF on from when they had their intracranial pressure monitors placed on our EVD, extraventricular drain. The problem is that in neurosurgery there's been a little bit of a migration away from using that approach, mainly because of a higher risk of infection of those catheters relative to an intraparenchymal monitor. And so that's a little bit beyond our control, but so we'll have those data on a mishmash of patients from the CSF. Same way, we have an ongoing phase 2B pediatric clinical trial that's prospective, randomized, double-blinded, and about half of those have an EVD in as well. So we may be able to piece together enough kind of more regional cytokine data versus the systemic.
Is that sustainable or a specific period of time that you measured and you believe that it's sustainable?
Is what sustainable?
The systemic anti-inflammatory action.
Well, so in terms of the cytokine effect, take cell therapy out of it. The cytokine effect has a couple of peaks, but it's a 24 to 120 hour post-injury event with various cytokines peaking at various times, which is why I wasn't very much enthusiastic at all about the whole CP thing, because I thought that's our time window. When it shuts, and that's our point of action, and forget about it later on. However, that cytokine story is one part of the story. Those affect the microglial / macrophage polarization, as do other influences. And so we think that really our target is the microglia / infiltrating macrophage, which have this much longer and broader inflammatory polarization. And so the influence on that early cytokine effect, yeah, I think that that's one important piece of the story. I think the microglial component of the story is this longer term readout, which we think is influenced by T regulatory cell input principally from the cell infusions that we do with the mononuclear cells. And that's really a numbers game. It's an immune system numbers game to get the kind of pharmacologic / biologic effect that we're seeing. And with the T regulatory cells impact on the microglia, that has a much more durable, sustainable response. That's our working paradigm.
Great, thank you.
Thanks to the audience for your time and attention at this session. Thanks very much to our presenters. Thank you.